is Chapel Hill, North Carolina, and her current research focus is intracellular pattern recognition, receptor involvement in inflammation, with Dr. Allen as her advisor. And her fun fact is that she has met and has been to Shaq's house when he was still playing Boston.
Yeah, it's kind of a crazy story. So, first of all, thank you guys for coming. Dr. Allen sends his apologies. He had a last-minute meeting that came up, so he will be unable to visit with us. But yeah, my project right now is really awesome. I'm really excited about it. There's one aspect of it that I'm not going to be able to share with you today, but unfortunately, we'll talk about that a little bit later. So the title of this talk is Inflammation Regulation and Inflammatory Bowel Disease. And to give you a little bit of context, this is going to be annoying. Okay, it was just working. Sorry about that, guys. But it's not hooked into the computer. It's like this. So anyway. So, when nothing is working, everything is frozen. Sorry.
Dr. Wong is normally an expert.
Oh, great.
There you go.
Awesome. My intro today. Okay, so bacteria are a normal part of the gut flora. We actually need some of these commensal bacteria to help us in like digestion, etc. So such that they occupy the gastrointestinal tract, such that nature actually used this as a cover for one of their journals that came out, our other genome. So bacteria are occupying every aspect of our body. There isn't a single surface that isn't covered or that has some sort of bacteria involved in it. The brain is something different. However, the picture on the left is just a breakdown of how many bacteria are occupying each of the sections of the gastrointestinal tract. So there's a whole heck of a lot, okay? So it's about 10 to the 9 in our gastrointestinal tract of the different types, the quantity of bacteria, over 500 different species. So things are not working here. Okay, there we go. So lymphocytes are basically, lymphocytes are about 50 times 10 to the 9, but our bacterial species are about 10 to the 14. So how are we regulating this quantity of bacteria in our gut? So inflammatory bowel disease is kind of a reflection of the enormity of the bacteria that we're trying to regulate in this region, okay? So you can get inflammatory bowel disease from stress, your diet, different chemicals, which is actually something that we use for our model. But there are a couple of diseases that are really prevalent in inflammatory bowel disease, which is not to be confused with irritable bowel syndrome. Inflammatory bowel disease is really characterized by the inflammation that is in the gastrointestinal tract region, whereas irritable bowel syndrome is just more or less dysregulation of the contractions of the tissue. So Crohn's disease and colitis are really the main two that people focus on when we're talking about inflammatory bowel disease, but there are others. And actually, Dr. Allen has been reaching out to other people working with, for example, rotavirus, because rotavirus can actually cause a lot of diarrhea, etc., and especially in underdeveloped countries, but that's a talk for a different day, I think. But inflammatory bowel disease is thought right now to affect about 200 in every 100,000 people worldwide. So it really is something that has interest for a lot of people. I think regulating the CDC says that the United States alone spends like $1.6 billion in patient visits for IBD. So how is the immune system kind of regulating all of these microbes and the immune cells? Because again, you have bacteria that are good, but at the same time, you don't want to let the bacteria that are bad overtake things. So this is a very broad schematic of what happens in the gastrointestinal tract, focusing on that just currently. but again, you have surfaces all over your body, so this is a little bit different, but the scheme is basically the same. In the gastrointestinal tract, you really have a mucosal layer that is protecting the epithelial cells, so that's like your first barrier. So then your epithelial cells, this is really gonna get wild. Your epithelial cells are equipped with villi, which yes, they increase the surface area, but you're really, it's a really thin layer. It's a one cell layer, and underneath that you have your lamina propria. So how are these things working together? All right, so again, you have your villi. You have Peyer's patches, which are regions in the body that are, I guess, how do I say this? There are regions in the body which are prevalent for the presentation of antigen by your dendritic cells to T cells, but we're not concentrating on that aspect today. Crypts are basically places where the epithelial layer, when it's damaged, they get replenished. So in your crypts, you have your stem cells. Lamina propria are fibroelastic kind of tissue layers underneath the epithelial, the epithelial layer, and then the mesenteric lymph nodes are places where the lymphatics kind of drain in this region. So in this region, the lymph nodes are basically another localization site for your adaptive and immune cells for presentation, but not necessary for what we're talking about today, but just to give you a general view of how things are regulated again in the gastrointestinal tract. So this is, I guess, more local view of the interior of a Peyer's patch. So these M cells are actually regions where antigen can transverse and get into the Peyer's patches. So dendritic cells can either sample antigen using their dendrites going through the layer of the epithelial into the lumen to, I guess, sample antigen to present to T regulatory cells to say, hey, this is okay, you can damp down your immune response. And in fact, that actually happens. So here is just a view of an antigen crossing the M cell into a macrophage, and the dendritic cell sampling antigen from the lumen to present to, it becomes activated, once it has sampled the antigen with the appropriate receptor. And then it can relay a signal to the T regulatory cells, hey, this is supposed to be here. And you have dampening with IL-10 cytokine release from the T regulatory cells of the immune response, okay? So this is kind of how things are regulated. So when things get out of hand, what's happening? You can have barrier disruption again from the several things that I mentioned earlier, stress, chemicals, etc. But when you have this barrier, when the barrier is destroyed, the epithelial barrier is destroyed, you have the activation of a lot of your immune cells. T regulatory cells are no longer secreting IL-10. You have effector T cells that are secreting interferon gamma, IL-17 and TNF, and neutrophils are recruited. So neutrophils are really kind of smart bombs. They get in, they destroy everything. So again, this is a large area, about 200 square meters of tissue in your gastrointestinal tract that's being regulated by the immune system with this very large quantity of bacteria. So you can see why dysregulation of this region and inflammatory bowel disease may be of interest to a lot of scientists. So when things are cleared, after the bacteria has been recontained, then you get, I guess, a reset button of your gastrointestinal tract with these players. However, how are you even sensing that these players or these bacteria are infiltrating the area when they're not supposed to anyway? So we have pattern recognition receptors on the exterior and interior of our cells, especially of our immune cells. But you may have heard of toll-like receptors. These are extracellular pattern recognition receptors that are sensing antigens. So toll-like receptors, once they are activated, can secrete or can activate the production of pro-inflammatory cytokines, such as IL-1 beta, which is really important for inflammasomes, but also IL-6. So, intracellular pattern recognition receptors. In Dr. Allen's lab, we work with nucleotide binding oligomerization domain nod-like receptors. So we'll just call them NLRs because that's a mouthful. So what these are is these are also pattern recognition receptors that are sensing antigen that are within the cell. So you've got extracellular and you have intracellular. What's important, I guess, about inflammasomes is that it requires activation from an extracellular ligand. So you need TLR activation to get the production of pro-inflammatory cytokines, IL-1 beta and IL-18 before the inflammasomes can actually begin their job, I guess. So, what is the job of the inflammasome? So the inflammasome formation, and there's a slide missing, so I'm going to have to explain this, I apologize for that. Inflammasome formation deals with the activation of pro-inflammatory cytokines, IL-1 beta, and IL-18. So what does activation mean? It means that the cleavage of those proforms of those cytokines are, I guess their maturation process, which is the cleavage of those forms, happens through inflammasomes. So you need inflammasomes to get the maturation of IL-1 beta and IL-18 and then the subsequent secretion, okay? So for NLRP1, there's about 11 different inflammasome forming NLRs, so there's more than that, but of the ones that we know, there's about 11 that form inflammasome complexes that are able to cleave the proforms of IL-1 beta and IL-18. So not much is known about NLRP1, and Dr. Allen had done some studies at UNC during his postdoc with inflammatory bowel disease and colitis-associated cancer models that implicated NLRP3 in those, as a protective role, in those disease models. So what we wanted to do is we wanted to look at NLRP1 because not much is known and really try to get an understanding of its role in colitis and colitis-associated cancer. So for today's talk, we will be concentrating on the colitis model. So we started with the hypothesis that NLRP1 also plays a protective role in inflammatory bowel disease through colitis only. So here's the model that we use. So we use dextran sodium sulfate, which is basically a detergent to induce damage to the epithelial layer in our mice. So we put them on a DSS treatment of water, which is we add DSS to their drinking water, and they will ingest the water for a period of 5 days. And after 5 days, we're changing the water back to normal tap water. But what's happening during this 5 days is the mice are drinking the water, but you're getting really severe, I wouldn't say severe, that's a bad word, but you're damaging the epithelial layer and allowing those bacteria to come in and cause inflammation. So we plotted their weights and we monitored the mice during this course. We monitor their weight 3 times a day. So we're weighing these mice 3 times a day to get an idea of how sick the animals are. So this is a survival curve which does not have anything to do with weight. However, this is really interesting data because of my gene of interest, NLRP1, we had a cage basically just so sick that they needed to be put down. So those were the moribund mice that really couldn't handle the stress of the DSS treatment. So in our knockout NLRP1 mice, they were not able to sustain the treatment and had to be euthanized. So that's one indication of how sick they were. But another indication was just their weight. So here we have, in the previous slide, we also had Caspase 11, but I'll explain that in just a second. So here we have knockout NLRP1 mice and wild-type mice put on the disease model. So we have control cages and we have DSS treated cages. And for both the models, the DSS treated cages are really crashing by day 6. And when I say crash, their weights are low. So we monitor their weight, and if they get above a 20% decrease in weight, we have to put them down. So as you can see, these mice are they're really approaching that weight where we have to put them down, but I think we kept them in the model with a close eye because we wanted to see like their recovery. So again, this is a significant difference in their weight on day 6 between the wild type and the NLRP1 genetically knocked out, model on the DSS treatment. So caspase 11. Previously, knockout models generated for caspase 1, which is the autocatalytically activated enzyme that actually does the cleavage of pro-IL-1 beta and IL-18. These, what they thought was a single knockout actually was a double knockout. So they ended up knocking out caspase 1 and caspase 11 in these previous models. So recently we've been revisiting, our lab and other labs in the field have been revisiting a true caspase 1 knockout and a subsequent caspase 11 knockout just to see what's, that dynamic, I guess. So in the caspase 11 mice, they're also not well. The weight is significantly decreased, again on day 6, in both the caspase and the wild type animals that were on DSS treatment, but the caspase 11 mice are continuing to crash by day 11, so they really don't begin to recover after the DSS treatment. So we also are monitoring the mice with a clinical score. This clinical score is based upon, sorry for those of you who are still eating lunch, but it's based upon their stool sample, if the stool is bloody, if the stool is I guess runny, it's not a well-formed pellet. We also score them on, excuse me for a second, weight is a part of it, stool, behavior, and condition. So if the mice are not grooming well, if the mice are not being social, then they will have a higher score, which would reflect how sick the animals are as well. So as you can see, the NLRP1 mice are clinically more sick than the mice that are untreated, the wild type mice that are untreated, and also the, the mice that are not on DSS and the mice that are on DSS. So they are showing signs or demonstrating signs of their sickness. Okay, so on day 6, we harvest the tissue for histology, and during that process, we are looking at the length of the colon. So what is that telling us? So as inflammation is occurring in this region, the tissue is actually shortening, and you can measure that, and we did that. And so we see that in a wild type animal that did not get any DSS, they have a longer colon, which you would expect. So the colon length actually is significantly decreased in the caspase 11 and the wild type, but I could not significantly attribute the decrease in NLRP1, and I believe it's because of the large error bar. So this error, So why such a huge error bar difference? It's a really good question. I believe it has to do with the starting weight of the animals, so we try to match them as much as possible, but this is a live model, and sometimes the weights are changing and you can't control that, but a larger weight would be a larger colon, and unfortunately our data here didn't really match up to tell our story, but you can see that there is a decrease. Okay, so of the cytokines of interest, we still have IL-18 to do, but we were able to get a good idea of what was happening with the cleavage and cleavage of IL-1 beta through the inflammasome or lack of inflammasome formation with NLRP1. So what we did is once we harvested the colon and measured its length, we I guess, dissected sections of the colon, distal and proximal, and cultured it in supernatant overnight. So what is that going to tell us? Well, the cytokine release from the tissue would give us an idea of how much cytokine production is actually happening in that particular tissue, in that section. So here we have distal sections of the colon averaged with the proximal sections, and the IL-1 beta production you actually have on wild-type mice, on the DSS model, you have an increase in IL-1 beta production, and you also have an increase in IL-1 beta production in the model that is a DSS, in the NLRP1 knockout model that's also on DSS. So this is interesting because you would think that, well, as a protective role, it is doing its job. It's showing that it's playing a protective role by suppressing the production of IL-1 beta, but it was just interesting because as an inflammasome, well, when you think about inflammasomes, you think about an inflammatory regulator in a negative way, not in a, in a positive way, not in a negative way. So for me, it was just interesting to see that this is another negative regulator, NLR, that we have at work in our immune cells. So moving on, we also looked at the production of IL-6. So IL-6 is also upregulated on the DSS model for the wild type and the NLRP1 knockout mice. IL-6 is a potent inflammatory cytokine. It's really demonstrating that, yes, in this localized region anyway, you have pro-inflammatory effect or function, I guess. In the caspase-11 mice, you see, again, upregulation of IL-1 beta and also IL-1/6 as well for the caspase-11 knockout mice in the same region. So what I wanted to show that I was unable to because I didn't have the time to put together those samples was some histology. So we also did histology on these samples to look at the immune cell infiltration into these regions, and some of the pictures that I have are just really gorgeous, but I wasn't able to put them all together for comparison purposes for today. But based upon the data that you have seen so far, we are able to say with a certain degree of certainty that NLRP1 does play a protective role in colitis in our experimental model, and yeah, that's kind of what we were expecting. So moving forward, what are we going to do? Well, we've been running a chronic version of the colitis model, which is basically looking at the progression of colitis to cancer, and its model is very well known and characterized and I guess published, but it's basically 3 rounds of DSS over a course of 9 weeks, and so that study came down in early January, but we're adding to it another study that Dr. Allen has not broken down just yet, so I was not able to present that data to you, but that's next for us, and looking further into NLRP1 as a therapeutic, especially again since those numbers for the cases of inflammatory bowel disease worldwide are so large. It is really an interesting, I guess, an interesting thought to look at the protein to see if it could be used to kind of suppress or attenuate some of those diseases. And then I guess what I'm most excited about is a collaboration that we have with Dr. Lindsay that I've been working with Jeff on. So NLRP1 currently has 3 known ligands, and one of them is T. gondii. And so what's interesting about this is, as a negative regulator, T. gondii actually shuts down the immune function. So Jeffter and I have been working to kind of look at that mechanism, NLRP1 and T. gondii together. So those experiments have been ongoing and have been really interesting. But hopefully I'll have some data from that to put together later this week. And I guess I just want to thank BT Prep IMSD, our, sorry about that apostrophe, but our track staff is really fantastic. They're great, taking care of the animals and letting us know something's wrong. I wanted to thank Dr. Lee, Bianca, and Matt have been really great with helping me understand toll-like receptor signaling and the balance, I guess, of cytokine or ligand stimulation for production of IL-6. Ahmed Lab, Catherine Cowan, she's been great with just everything, all my general questions, and then Dr. Allen and the rest of Allen Lab for helping to complete all of these experiments. And of course, the Virginia Maryland College of Regional College of Veterinary Medicine and Virginia Tech. And at this time, if you have any questions, I'd be happy to try to answer them.
So did you say that DSS shortens the colon? Like when you have,
So the chemical itself doesn't
I see.
shorten the colon.
But it's like-
What's happening is that DSS disrupts the epithelial barrier, which
I see.
allows the bacteria to get in and cause like a lot of inflammation. So it's the inflammation that's actually, I guess, think of like a balloon, right? Like when you add more water, that was a bad analogy. No, actually it's okay. Like if you have a balloon with no water, you can stretch it long, right? But if you add a whole bunch of water in it, you're not gonna stretch it as long as you can if it didn't have water in it. Think about it like that. So when you have all this inflammation going into the tissue, you're not able to extend it as far. Does that?
yeah, that makes sense.
Okay. Hi,
In your, that big error bar in your
Yeah.
NL1 knockouts,
yeah.
did you weigh the animals, each individual at the beginning and at the end?
We
I
absolutely
think you
did.
might be able to reduce that variability by expressing percent weight change for all the animals, because that would deal with the problem of inadvertently selecting animals that happen to be more variable in that group.
So you're saying being more specific to the animals that are the most sick then?
No, instead of expressing absolute weight at the end of the study, express end of the study weight as a percent of the weight for that individual at the beginning of the study.
So for each
I see what you're
You
saying.
see.
So,
yeah, it's a difference in the weight, so you're doing
Right.
the weight change.
So we do that actually
Oh, you
over-
did
Yeah,
that?
we do that for when we're tracking the weight, but when we're, I guess, currently the mode in publishing is not to have that data set that way. Does that make sense?
Well, true, and there are always conventions in fields, but
But it
when
can tell
you
their
get a
story.
result like that, you could try doing all your groups as, for each individual, it was, 120 or 110 or whatever of its original weight.
Right.
And if that eliminates the variability, then you can accept your explanation that maybe it was due to more initial variability in that group. And see, even if the convention is to express absolute weight, then you've taken it a step further and provided an explanation for how that result came about. I mean, you still usually need to replicate the stuff anyway, but that would add strength instead of just speculating that this is why it happened.
So I guess the population size is really small, so that for that particular group, treatment group, the population size was 6. So we do have to average this data with the other, other experiments that we'll be running for the colitis model in the future. But that's a great suggestion. Thank you, Alicia.
I have so this is not including the yogurt mice and
No.
the ones that they took off the assessment.
Yeah,
right?
so Alicia is referring to is our colitis associated model, which began well and started to crash like almost immediately. So basically what happened was tracks. So the track staff had added yogurt chips, which we joke in our lab are like white chocolate Hershey Kisses. And so the mice became dependent on these yogurt chips. So when we took away what became their primary source of food and then put them through the DSS model, it was way too much stress, and we had mice like coming down like it was an absolute nightmare, such that we had to explain ourselves to everybody. So that will be data that will have to be explained very, very delicately when I start to talk about the CAC model. But no, it didn't have any relevance to the colitis study.
And I think you told me the other day that caspase is a cleaving enzyme that cleaves the proform of.
Yeah, so these caspases are cleaving pro-IL-1 beta and IL-6. Not all caspases cleave pro-IL-1 beta and IL-6, but caspase 11 and caspase 1 are known to do this, and caspase 11 is also known as caspase 5. So
Are you-
reading through literature.
So are you planning to do a protein lot? I can't remember if that's Southern or Northern to show.
Western.
Is it a Western? Is that the protein one? Okay, I can't remember.
It's okay.
So do you see a difference between the pro and the final form, the cleaved forms on your blocks?
Yeah, so that's actually something that's a great question. IL-1 beta is a very potent cytokine. It's not secreted in large numbers. We are having a very difficult time getting a Western blot of IL-1 beta in either its pro or cleaved form at this time. I've been working with, again, Lee Lab, they're so awesome, with those guys, and I think that we've got a better idea of how we can make sure we get images to happen, but that is something that's on the books that we need to have happen
Okay.
for what we're putting out this summer. So
I didn't know if you've done it yet, because I'm having
I've done it
issues
like
as
15
well,
times.
showing the protein fold expression
Yeah.
versus the actual presence of the protein.
yeah, because no one can
So-
find it. No one in
Okay.
vet med can get a Western of IL-1 beta. So if any of you have any ideas, please hit me up and let me know.
People ever use immunohistochemistry to look at it?
Yeah, so Dr. Allen is so full of great ideas, we just haven't gotten around to do it. So it's something that we've talked about. We've talked about mass spec, we've talked about a couple of other different things. But at this rate, we've been traveling a lot with interviews, and he's been traveling a lot, so it's just a question that's gonna happen this Friday at the next, lab meeting. We're gonna figure out how to move forward with that. Any other questions?
What's wrong? Just hit escape.
Okay. I'm trying to get some of these. Let's escape it. Oh. The mouse is, okay, there we go. I'm sorry.
What's wrong?
Mine popped up again, even though I pulled up.
What is it for?